Hello everyone, my name is Mohit Verma. I am a postdoctoral fellow at University of Missouri, Columbia. I'm going to present on elucidating the mechanism of SA-4-1BBL immunoprevention through advanced informatic approaches. So SA-4-1BBL train the immune system to prevent cancer by activating targeted long-lasting T and NK cells responses with fewer side effects than 3S3, so making it a promising cancer vaccine candidate. So as a new paradigm that has emerged, the cancer immunoprevention is achieved through promoting anti-tumor immune surveillance to block tumor formation and progression. Two different types of tumor can be targeted by immunoprevention: tumor caused by infectious agent or the one caused by non-infectious agent. Effective vaccines are available for viruses associated with cancer development. At this point, immunopreventive approaches for the tumor of non-infectious agents are at preclinical level of development. So our approach to finding a solution to combat cancer by preventing this internal danger on discovery of novel immunoprevention pathway and immune targets, preclinical development and testing of interventions, and investigating mechanism of efficacy for precision cancer prevention. So SA-4-1BBL alone provide robust, long-lasting cancer immunoprevention across tumor types, unlike the 3S3 antagonistic antibody, despite potential anti-drug antibody concern. This is a workflow we followed for bulk RNA-seq analysis. And this slide represents the RNA-seq sample metadata showing high quality alignment, which is greater than 96% across all the samples. In this, we consider 49 RNA-seq comparisons groups categorized by treatment tissue type, like injection site, lymph node, injection frequency 1x, 2x, or time points D3, D7, providing a comprehensive framework for evaluating differential gene expression across all experimental conditions. This slide represents a comprehensive summary of immune gene expression pattern and treatment comparisons. Panel A is a schematic of experimental design and sampling timeline for bulk RNA-seq at both lymph node and injection site across days and time points. Panel B shows the PCA plot showing clear separation by treatment, tissue type, and time points, confirming data reproducibility and distinct transcriptomic signature. This table summarizing differential gene expression in lymph node and injection site across treatment and time points, and SA-4-1BBL shows higher differential expression, the gene counts, and more unique transcripts. If we talked about the differing trend between 3S3 and 4-1BBL, this highlighting the robust and divergent immune signaling, especially in injection site samples. So Venn diagrams illustrate gene overlap across treatment, tissue, and conditions, with SA-4-1BL showing broader and more sustained gene activation profile than 3S3. So after differential expression analysis and based on trend analysis, we characterized genes based on k-mean clustering, and 8 distinct cluster revealed is gene regulations, and some of them are IS specific, some of them are LN specific, specifically like cluster 2 and cluster 5. Those regulatory genes like TNFY3, IRF3, and COL1A2. So we characterized by using the barcode and strategy, introduce a 6-digit barcode system, stratify differential gene expression by tissue, time, and injection site. And we also identify 131 unique expression barcode. And especially cluster 3 represent more muscle contraction genes. Over here, you can see the smooth muscle contraction and sustained muscle contraction. So importantly, SA-4-1BBL selectively induce chemokines and cytokines, promoting immune recruitment without triggering somatic toxicity. Transcriptional comparison confirmed that 3S3 drive rapid activation of innate pathway, C-type lectin receptor, toll-like receptor signaling, and associated network suggested a role of pathogen defense. So in contrast, SA-4-1BBL promote adaptive regulation through Th17 differentiation, cancer-related pathways, apoptosis, and inflammatory disease signaling. So IMPRESS analysis, after doing the random forest ranking, we selected the 27 trend differentially expressed genes to perform the IMPRESS analysis. So in this slide, these 27 genes were analyzed to impress, and SA4-1BL drives adaptive immune activation by key regulators like STAT1, CD247, and IFNG enhance TCR-NK pathways and tissue remodeling with sustained effect over time. In contrast, 3S3 primarily activate innate inflammatory signaling with limited pathway overlap, indicating distinct immune strategies. So this slide summarizes the 8 k-mean cluster, each with a distinct tissue-specific temporal and treatment-responsive immune gene signatures, highlighting the SA-4-1BBL dominant role in adaptive and innate immunity modulation. So, in conclusion, the identification of CD151 TGF beta axis introduced a novel mechanistic insight with therapeutic implementation suggesting new targets of enhancing immunopreventive efficacy. Notably, SA-4-1BBL modulates pathway associated with ECM modeling and cell proliferation, while 3S3X exert effect primarily through receptor level activation and acute inflammatory signaling. So SA-4-1BBL elicites broad, sustained, and tissue-coordinate immune response, but distinct gene and receptor cluster uncover the mechanism of adaptive immune versus inflammatory stress. So findings support for SA-4-1BBL as a promising immunotherapeutic agent for cancer immunoprevention is a very important thing in this project. So integrating of clustering, IMPRESS, and pathway analysis offer a deep understanding of treatment-specific immune dynamics. So the take-home message from this topic is the discovery of additional immunopathway through advanced informatics may pave the way for the universal and development of cancer vaccine. We also developed a cancer immunoprevention resource. And here is the link. You can go and visualize all the plots and play around with the data. And we are trying to integrate the single cell and our bulk RNA-seq overlay with this work and enhance the visual analytics of hypothesis testing and biomarker discovery. In the end, I would like to acknowledge the BBME department colleagues, my supervisor, Dr. Tsuyoshi, and all other colleagues, and immunomodulation and translation research program colleagues, Dr. Hubble, Shimon, Faiza, and others. And I would like to acknowledge our funding agency by NIS, DoD, and KSTC, and thank you so much for listening.